Okay, so this voiceover to take the place of lecture on Friday, April 1st is going to continue talking about the cell cytoskeleton and cell movement from chapter 13. So we talked about various forms of actin filaments and protrusions that can come from the cell. We talked about microvilli right before the end of class. I want to talk about protrusions that aren't permanent to the cell like the microvilli are for epithelial cells, but protrusions that are more transient. And so here's some examples of a couple types. So we have the pseudopodia are protrusions. These are what some organisms have for movement. This is pseudopod. It's kind of like pseudo is like a kind of like a fake foot almost. And so these are forms of movement where there's transient protrusions from the cell that are there for the purposes of movement, where a transient protrusion, actin filament, will extend outward from the surface of the cell, make contact with a solid surface, and then actually use that to pull and move the cell in a particular direction. There's also lamellipodia and filopodia, and so this is showing examples of that. So L here is showing where the lamellipodia is present. So lamellipodia is going to be the more kind of widespread protrusion, whereas filopodia is more of a single tiny bundle protrusion. So lamellipodia is going to be more like the actin networks that form. Filopodia is more of an actin bundle, a single thinner protrusion coming out of the cell. And again, mostly these are going to be involved in the process of movement. Transient protrusions are. Alright, so the next couple slides are going to have a couple questions. What I'd like you guys to do is answer these questions on the discussion forum of the portal. So I'm going to put a forums post up, and I'd like you to post your answers to these questions. It'll be a classwork for April 1st, since we don't actually have classes, so I'll grade you on classwork. And so if you could do that and have these posted by, let's say, 11:55 p.m. on Sunday, April 3rd, then we get full credit for that particular coursework today. So this is talking about true or false. Shortly after polymerization, the terminal phosphate of the ATP bound to the actin monomer is hydrolyzed, and the resulting phosphate and ADP are released from the polymer. So talking about the treadmilling process of actin, decide whether you think that is true or false. Cytoskeleton prevent actin from polymerizing by binding to the barbed end of actin filaments. Phalloidin stabilize filaments against depolymerization by binding all along the side of actin filaments. So what do you think that is true or false? The actin filaments in the lamellipodium of a moving cell appear to be more organized than they are in other regions of the cell cortex. Many of the filaments project outwards in an orderly array with their barbed ends inserted into the leading edge of the plasma membrane. I just talked about lamellipodia a little bit, and so just take your shot at whether you think that is true or false. If not, we'll talk about that more in class on Monday if we need to clarify anything. The actin bundling proteins responsible for parallel bundles, contractile bundles, and networks each have two actin binding domains, and all the actin binding domains are related. I think that's true or false. The main transmembrane linker proteins of focal contacts are members of the integrin family, whose external domain binds to an extracellular matrix component, while the cytoplasmic domain is linked directly to actin filaments in stress fibers. True or false? True or false, at the base of the microvillus, the actin filament bundle is anchored into a specialized region of cortex known as the terminal web. And I'd like you to fill in the blanks for this one. Blank are finger-like extensions found on the surface of many animal cells. The bundling protein blank, which is found only in microvilli, cross-links actin filaments into tight parallel bundles. Alright, let's talk a little bit about how actin is involved in the process of cell movement. So let's talk about myosin. So myosin is basically a molecular motor. And so what myosin does is it's going to interact with actin filaments and convert chemical energy into mechanical energy. So take the chemical energy that the cell has accumulated through all the metabolic reactions and convert that into mechanical energy that will actually allow the cell to move. So muscle fibers are one of the big areas of the body, if you think about the human body, that is going to be using a lot of myosin. It's a lot of movement for muscle cells. And so muscle fibers form muscle cells, and muscle cells are basically large cells with multiple nuclei that kind of come, like multiple nuclei in one large long cell that come together to then form muscle fibers. So the cytoplasm of muscle cells is filled with myofibrils, and myofibrils are bundles of actin and myosin together. And so each myofibril that's present in the cytoplasm of these muscle cells is a chain of contractile sarcomeres. And so it's going to be a chain of something known as a sarcomere. Let's take a look at the structure of a sarcomere here. All right, so this is showing the muscle cell structure. So here we have our muscle. And so in muscle, they're in bundles of muscle fibers. And so one muscle fiber, if we think of one muscle fiber as a muscle cell, which we can, you see we have multiple nuclei, so there's multiple nuclei in a muscle cell. There's a plasma membrane to the muscle cell, and in the cytoplasm of the cell, so if this whole thing is a muscle cell, in the cytoplasm we have what are known as myofibrils. And so the cytoplasm membrane around it, and then the cytoplasm is full of these myofibrils. And each myofibril contains a region known as the sarcomere. And so a sarcomere is going to look basically like this. So it's going to extend from the Z disc to the Z disc. So it extends from the Z disc to Z disc, and it has this other portion known as the A band. So look at what that's doing. Okay, so let's look at the structure of a sarcomere. So for a sarcomere, we're going to have two discs and lines we should be aware of. So we have what's known as a Z disc here, shown in green. The Z disc is going to be the portion where actin is extending outwards. So a Z disc is going to have actin filaments extending out from one side as well as the other. And we also have an M line, which is going to have, instead of actin, it's going to have myosin extending out of both sides. And there's going to be regions of overlap between this actin and myosin. And this region of overlap, the interaction between actin and myosin, is what's going to allow the conversion of chemical energy in the form of ATP into mechanical energy in the form of a contraction, in this case, movement of the muscle fibers. So if we look up here in the electron micrograph, we have an A band. So the A band basically it basically covers the width of how far the myosin filaments extend from the M line. So myosin extends from the M line out to a certain point. That's one edge of the A band, and that goes all the way to the other edge of the A band where the myosin filaments extend to. And then in the A band, there's going to be a portion known as the H zone, and the H zone is the portion of the A band where we don't have overlap between myosin and actin. So it's a portion where only myosin filaments are found. It's a little bit lighter than the rest of the A band, which is darker, showing the overlap between the actin and myosin filaments. The other band is known as the I band, and the I band is basically the portion where we only have actin filaments present with no myosin. So the portion of actin that is not overlapping with myosin is going to form the I band. Okay, so the model for muscle contraction is what's known as the sliding filament model. And on Monday in class, I'll actually bring in sarcomere to show this model in great detail. But what I wanted to just kind of briefly run through it here. So the idea of the sliding filament model is that during a contraction, what's going to happen is the Z disc, and I should point out that when we have the Z discs, the barbed end is attached to the Z disc, and the pointed end is attached away from the Z disc. Just, orient yourself to what you already know about the structure of an actin filament. So what happens during a muscle contraction is the Z discs are going to do sliding. So they're sliding of the Z disc and the filament sliding towards each other. And so this contraction is actually going to shorten this length from one Z disc to the other. So we're going to have a shortened length of the Z disc. That leads to this muscle contraction. So these are going to slide together, forming this muscle contraction. So when that happens, the region of overlap between the actin and myosin basically comes as though they're completely overlapping with each other. So there's no portion of the myosin or actin that are not overlapping at this point. So we're going to bring our Z discs together as far as they can, Z discs together towards our M line here in the middle, and that's what's going to allow for muscle contraction to occur. So it's a sliding that then allows muscle contraction to occur. Okay, so let's look at the structure of myosin. So the myosin that's found in muscle cells is mainly myosin 2. There's another form of myosin I'll talk about later. So myosin 2 basically contains what's known as, it's kind of basically a dimer. So we're going to have a portion where you have two heavy chains. So we have two heavy chains here. Kind of looks like a really long To be frank, it looks kind of like a long sperm molecule. So we're going to have 1, 2 long-looking heavy chains that are going to wrap around each other in a coil of 2 helical tails. So a coil-to-coil interaction, if you will. This is the 3rd time that we have seen such a structure. If you can name the other 2 times, that's the other thing I want to do on the portal. Name the other 2 instances where we've shown coil-to-coil domains of this kind of similar coil-to-coil structure that we have here for myosin 2. And so the heavy chains are what are going to wrap together to form this coil-to-coil structure. And then around we have the globular head regions of each heavy chain around what's kind of, for lack of a better word, the neck region. So the portion of the heavy chain, not quite the globular head, but before it gets to the coil-to-coil domain, we're going to have 2 light chains wrapped around each other. We're going to have what's known as an essential light chain and then a regulatory light chain. So this is going to form 2 other portions, and this is going to be for each of the 2 heavy chains is going to have this lighter chain wrapped around it. Alright, and so myosin filaments are thick. So thick muscle filaments, there's basically several hundred of those myosin molecules I just showed in the previous slide, arranged together in a parallel array. So if you look at our M line here, extending outward, we have about several hundred myosin filaments arranged together in parallel with each other. Basically kind of like golf clubs coming out of our M line here, if you think about it. A lot of golf club looking things coming out of our M line, arranged parallel. And this globular head portion of the myosin structure is responsible for actually binding to actin. So the globular head of myosin is going to actually attach to actin. So here you'll see our actin molecule extending out from the Z disc. Other things you should be aware of, and we'll talk more about the function of these on Monday, is we have a molecule known as titin and also nebulin. So we'll talk about those on Monday, what the purpose of those are. And again, just orient you, the barbed end of actin is attached to the Z disc. The pointed end is going to be attached away from the Z disc. And so what happens is motor activity is going to move the myosin head group along actin. So when the contraction of the sarcomere is occurring and these Z discs from either side are going to come towards each other, what's going to happen is the myosin head groups that are attaching to actin are actually going to move further along the actin molecule towards the barbed end or towards the Z disc. So that motor movement, the myosin actually is going to move along the actin molecule. And that's what occurs as actin filaments slide towards the M line. We get movement of the myosin heads along the actin molecule. Alright, so this is showing a model for how the actin of myosin actually works to lead to a muscle contraction. Okay, so if we have here, and we start, we have a myosin globular head attached to a portion of actin molecule in the actin filament. And so if we have something that's going to stimulate muscle contraction to occur, we're going to have energy involved. And so ATP is going to come along and actually lead to dissociation of this myosin molecule with actin at that particular part. So it's going to dissociate the complex, which is important because remember the myosin is going to have to slide across the actin. So we have to allow those head groups that's already attached to dissociate. And so ATP is going to come along, and this hydrolysis of ATP is then going to dissociate the myosin. So when ATP is hydrolyzed to ADP, that's going to result in a conformational change of the myosin so that it's going to dissociate from the actin. And when it does so, the movement of the myosin is actually then going to bind to a new position on actin. And there goes our phosphate away. So now I have myosin bound to ADP at a new position on actin. And then what happens is myosin is then going to return to its original position as ADP is hydrolyzed. So as ADP is hydrolyzed off, that's going to lead to the myosin filament now leading to its new position. And by the nature of the myosin filament moving to its new position, think of it as it's kind of, it's cocked in this position. When it moves to its new position, that movement is actually going to slide the myosin molecule along the actin filament. And this is what's known as the power stroke. We're going to actually have sliding, boom, of the actin molecule, of the myosin molecule on the actin filament, which is going to lead to the actin filament sliding when the myosin reverts to its original position. It's still going to be attached to that same piece of actin. It's just going to, the conformational change that occurs is going to slide it along the actin filament. And this is how myosin leads to movement along the actin, or sliding of the actin filaments. And this is what leads to muscle contraction occurring. Alright, so what is going to happen to stimulate muscle contraction in the first place? So what happens is nerve impulses are going to stimulate the release of calcium from the sarcoplasmic reticulum. And when that occurs, we're going to have an internal spike in the calcium concentration. So it's actually going to go from a level of 10 to the negative 7 to 10 to the negative 5. So, a twofold magnitude, 100-fold increase in the internal calcium concentration levels that occur. Calcium is then going to bind, and that's going to prevent inhibition of myosin binding by what's known as the troponin-tropomyosin complex. So calcium binding, because the increase of calcium prevents the inhibition of myosin by the troponin-tropomyosin complex. So myosin is not inhibited, now myosin can allow to occur. All right, so here if we look at the calcium stimulation of muscle contraction, so here's an actin filament. Of course, I have to tell you that. You know what actin looks like on the little yellow actin filament molecules, polymerized together to form a filament. And then wrapped around actin, we have our tropomyosin. And we talked about tropomyosin already as a actin-binding protein. And so at all the tropomyosin, we have what are known as the troponin complex. And so the troponin complex contains three portions, troponin I, C, and T. And those are all going to interact with tropomyosin and actin. When calcium comes along, so the troponin complex is binding tropomyosin in such a way that this would be an inhibition of myosin because the myosin binding sites of actin are blocked in this situation. When calcium comes along, calcium is going to bind to our troponin complex, and it's going to bind in such a way to alter the structure so that now we're going to have the binding sites on actin of myosin exposed. So the binding sites for myosin to bind to actin, which are shown here with these little dots, little circles, that's going to, that now becomes open, which is going to allow myosin to now come and bind to the actin binding sites and stimulate for muscle contraction to occur. So it's this release of calcium by nerve impulses that alters the troponin complex and then allows the myosin binding sites to be revealed, allowing myosin to bind to actin, leading to muscle contraction occurring. And I mentioned in class on Monday that the troponins are involved as one of the ways that you can actually detect whether an individual has a heart attack by measuring the levels of troponin. So I want you to do some research or just think and also post on the portal on your answer sheet or the discussion forum due on Sunday, why would the troponin complex stimulate that someone might have had a cardiac event? So why would troponin levels be higher in an individual, let's just say troponin levels in the blood, be higher in an individual that has suffered a heart attack? So think through that, knowing what the heart does, what the heart is, why that would stimulate that a heart attack has occurred. All right, and this is just showing in a little bit more detail the interaction of calcium with the troponin. So here we have our troponin complex. Here's tropomyosin bound to actin, and so tropomyosin in an inactive cell, a cell that's not being stimulated for muscle contraction to occur, is going to have tropomyosin blocking the myosin binding site of actin. Calcium comes along, binds to troponin C, so calcium specifically binds to troponin C, which is important. That's where the name of C actually came from for troponin. Calcium binds to C, and that's going to alter the complex of the structure such that now the myosin binding site of actin is clear, allowing our myosin head to now bind to our actin molecule. All right, so I'm going to go and stop there. I'll talk about non-muscle cells in class on Monday. We'll hopefully finish up chapter 13 in class on Monday, April 4th. So please do remember to post the answers to the questions that were proposed in this PowerPoint by Sunday at 11 to 5 p.m., and we'll discuss it more in class on Monday.